Okay, hi, so this is going to be a revision video for D1. The HL content is very interspersed with the SL content for this topic. It's not as standalone as it is in some other areas, particularly for this first area. So I'm going to kind of try to just basically go through it at an HL level, but I'm going to indicate every time it's an HL only content. So if you're SL, you may just have to kind of tune out for a second or kind of quickly fast forward because it's going to be a little bit less discrete, but that's just the nature of the topic that we are looking at. So starting with DNA replication, and again, this one, SL has to know fairly minimal about the process, but you have to know all the applications in terms of PCR and electrophoresis. HL has quite a bit more in terms of the enzymes in the process. So let's start with what everyone needs to know, which is kind of the purpose, the nature, and then the process. So those are kind of our background information. So we'll start with kind of what is the purpose of DNA replication. So hopefully that's pretty obvious, but kind of inbuilt in that is I want to make sure you know when we need to do this. So purpose of replication is to produce two identical copies of DNA. And not just a gene, but of the entire genome for that organism. So of basically all DNA. And then, like I said, going with that is why. Like, why do we need this entire copy? And we're going to do that anytime it's before cell division. So produce identical copies before cell division. And then embedded in that before cell division, because we haven't done division yet in the sequence we're going, is let's just check in and make sure we know when are the two times that cells divide. So that's going to be anytime we are growing and repairing. And then that's also going to occur when making sex cells for reproduction. So essentially leading up to either of those two events, we're going to need to actually replicate the DNA in order to prepare for the cell to divide. Should already know these terms, but we'll just add, when we say growth and repair, we mean before mitosis. When we say making sex cells, we mean before meiosis. Okay, so that's kind of our emphasis, and again, keyword here being identical, and then the other key thing is, do you know when it occurs? Next thing we need to know is what is the nature of it? Which by that, I'm just coming up with a reason to say like, what form of replication is it? And that's your vocab word that you need to know, which is semi-conservative. So it is a semi-conservative process. It is not in the course that you need to know how they discovered that. So the Messelson-Stahl experiment is not named in your syllabus anymore. So you don't need to know kind of the details, but it is empirically backed up by kind of using isotopes of nitrogen to track kind of where that original DNA ended up to be able to see that kind of division process. So when we say semi-conservative, we mean that each original strand of DNA and I'm just adding the word separated to clarify that, is going to act as a template to create a new complementary strand. And so one of the themes that you'll see throughout this is this idea that all of the processes in D1 are all coming about as a result of complementary base pairing. So that's going to kind of be a recurring theme, is that complementary base pairing is how we do this. And the idea that complementary base pairing allows for processes like replication and transcription is thought to be kind of the whole reason why DNA works in that way. So each original strand acts as a template for the new strand. If we do just a little tiny little kind of diagram, so we would basically say, here is my original DNA. It is then going to be split open and then each new strand is going to be copied across from that. So when we say kind of semi-conservative, we're referring to that idea that each of these are going to be half from the original and half are going to be new. And so remember, we did talk in class, there was kind of debate whether it was conservative, dispersive, and experimental data supports that it is in fact semi-conservative. So don't need to know the data, but it is good to know that it is backed up by it. Okay, and then what is the process? So we've said the purpose, we've said the nature of it, then the question becomes, well, how does that actually happen? So what is the process itself? And so the process is quite complicated. So we are going to say that it is a complex enzyme-mediated reaction that is kind of complicated due to the directionality of DNA. So that's kind of our overview statement. Then we'll go into kind of what are the steps. So it is complex in all organisms. So remember, it's so complex that we are going to go through the prokaryotic model just because of how complex it is in eukaryotes. Very similar, but even more complex. So it's complex. It's enzyme mediated, meaning there's enzymes involved in each step. And the reason why it is complex, so we'll say complicated by the directionality of DNA. So if you were kind of writing about it, the two things that we're kind of drawing out here is the idea that it relies on enzymes. So we want to remember that it's very enzyme driven, and that the reason for kind of the complexity of it in terms of leading and lagging strands for HL is due to the directionality of DNA, making it complex. Okay, so then what we're going to do next is we're going to do a little mini chart. And it's going to have to be like, if you're SL, you only need 2 of these 6 steps. So yours can be much smaller, or you can write bigger. If you're HL, I need you to kind of probably write pretty tiny because we still need to fit PCR and electrophoresis on the bottom of the page. If you are going like me for the mad mission to fit it all on a page, if you don't care about that and you just want to take as many pages as you want, you just spend all your space. I just have a neurotic thing about wanting single page summaries. So I'm going to erase up there just because I want to try to fit it all on one board. So again, that we are going to go through what it looks like in prokaryotes, simply because of the complexity in eukaryotes. So we're going to try to kind of take that into account. Okay, so we're going to make a note that these are going to be my steps in prokaryotes. And this is acting as a model. It is the same basic idea. The main complexity is the polymerases and so forth. There's many more of them, and it differs more by each different eukaryote. So it's not quite as one size fits all. Okay, so like I said, we're going to make a little chart. So I've broken it up into essentially 6 steps for HL, and like I said, SL, you really only need to know 2 of them. So you can kind of skip a few. So we'll go step, and then we're going to say, the step is just the number. So just make a tiny little column. Then we're going to say, what is the process? And then I want to clarify specifically state what enzyme is involved in that step. And then we're going to draw a little picture. So I'm doing the picture a little bit differently because in class, we already drew the big picture one. So we already drew the picture that had like everything on one picture. So just to try to do it a little bit differently, part of my goal in this video is to try to give it step by step, because if you're writing about an ERQ, you'll need to try to write about it in sequential order. So it's just a different way of presenting the same information, but we're gonna do kind of just a mini diagram that shows what's going on at that step, since you would never have to create a massive diagram. So we'll kind of go with it this way. Okay, so step 1, and everybody needs to know this. So step 1 is we need to divide the double helix into 2 single strands. So we need to break the hydrogen bonds in the double helix. To create 2 single strands. And so again, just a reminder that this does not happen through the whole thing. So this is going to start at what we call the origin and moves along. And that moving along is creating a replication fork. So embedded in this, I just have a bit of vocab. So everywhere I have some kind of vocab, we're just going to draw it out. So kind of vocab that we want to add is this origin, which is where it starts, and this idea of a replication fork. And kind of your key information that you would need to describe is that you would need to say how it happens, which is by breaking the hydrogen bonds. The enzyme that does this is helicase. There are supporting enzymes, so it is no longer in your course. You have to know gyrase and the single-stranded binding protein. So if you're looking at old diagrams or diagrams online, you will see those a lot of the times, but you only need to know helicase by name. So here is my original strand, looking like this, still double-stranded, and helicase is actually a ring that basically moves through one strand, and as it moves through, it's going to cut those hydrogen bonds. So that is my helicase enzyme there. Just to label the other 2 things, so this is where it would have made the first cut, so this is the origin. And where it is currently found, this area right here is the replication fork. So I probably should have pointed that not directly at helicase, but in the place being broken up. So where we are currently dividing, that is the replication fork. Okay, so that's enzyme 1, helicase. Next enzyme, HL, you're going to take a break. I actually think it's helpful to know about this, so I would recommend listening but not writing, because you're going to have to talk about primers and PCR. So to me, it helps to understand what they are, but you don't have to know, you wouldn't have to describe this. So step 2, at any time we have a HL only, I'm just going to put a little star and put HL. And that's just a reminder of that. So the problem is once they're split, DNA polymerase, which we'll get to in a minute, has the limitations that it cannot start a new strand. So it cannot just create the first base. It can only add to an existing strand. So in order to start the new strands, we're going to need primers. And in replication, these primers are made of RNA. So we are going to have the next step is that these complementary, that's going to be important every time we use that word, RNA primers are built and placed across from the DNA at the origin. And the purpose of this will be to start new strands. So we're going to place the primers there, and they are going to be complementary. And I'm also going to make a note that they are placed anti-parallel. And I'm going to try to draw that on the drawing, but that's going to have an implication as we look ahead and see kind of what that's going to do. But that's going to change kind of how our replication, the leading and lagging. So that's why I'm including that information. Okay, so a key concept is that they are RNA primers. So that's kind of a key thing that we want to know. We want to emphasize always that they are complementary and understand their purpose. So there isn't the enzyme that is involved in doing this. It's kind of strange because it's called DNA primase. even though it makes RNA primers. So it's building RNA primers for DNA replication. So you can just call it primase, it's fine. But that is officially like just to clarify when they're used for. Okay, so now let's draw our little picture. And this time it has split open a little bit more. Again, this is my origin. I'm gonna add some 3' and 5' now because it's gonna start to be relevant. So we just wanna clarify that. So let's say this is my 5' to 3' strand. This is my 3' to 5' strand. So when I say that the primers are anti-parallel, here's my little primer that's placed. So if this is a 5 to 3, that means this little primer is 3' to 5'. And this little primer is also anti-parallel, so it's gonna be 5' to 3'. Okay, so there's my initial step, is to place the primers. Again, SL, you don't have to know the enzyme or that they're RNA primers, but you do have to understand the role of primers in PCR, so it could be worthwhile. Okay, step 3, this is an everybody step. I'm going to use the terminology leading strand. You don't technically have to know if you're HL, so you can just say this is how it's done. I feel like it's almost so simplified it helps even at an SL level to understand leading and lagging. But anyway, see where your comfort level is. So what I'm going to do now, I'm actually going to have a 3 and 3A because they're happening at the same time. So it's concurrently, it's going to start the leading and lagging, but I'm going to put them as separate steps because it's quite unique. So we're going to build the leading strand. So we're going to say the leading strand is created. Is copied. So let's go through step by step kind of what I mean by that. So first of all, when you look here, DNA polymerase can only add to a 3' end. It builds phosphodiester bonds. They have to attach onto a 3' end. So the primer, for it to kind of be successful, the primer needs to end in a 3'. So like that primer, cha-ching, happy timer. So leading strand, the primer is 5' to 3'. The primer is, so it ends in the 3', which is great because that's what DNA polymerase can use. So then DNA poly 3, so I'm just going to abbreviate it for right now and call it poly 3. Poly 3 adds complementary bases, so complementary to the template. And bonds them to the free 3. Same previous base, previous nucleotide, you know what I mean. I'm just using the easiest word. Bonds it, perfect. And then just a kind of summary, because this is the leading strand, it's copying continuously towards the replication fork. Okay, so the enzyme here, and this is going to be the enzyme kind of in the next kind of step as well, is DNA polymerase 3. And again, this is where it's important that this is our prokaryotic model. In eukaryotes, there's different polymerases, and there's not necessarily one used for everything. But in this model, we're using DNA polymerase 3. And I'm going to also just add a little note that it also has proofreading abilities. So it has magic proofreading powers as well. Okay, so over here we have my diagram again. And so notice this is the one that we are using. So on this strand, because this ends in a 3', that is good. Cha-ching. That makes DNA polymerase happy. So then it is going to start adding bases from there, and then it is just going to keep going. So it's just going to build that continuous one, and my enzyme doing that is DNA poly 3. So there's my leading strand. So if you're an SL, you don't have to use the word leading strand. You can basically, for SL, just focus on this little line. So DNA polymerase adds complementary bases and bonds it to the previous base, building a strand. Ta-da. All right, so SL, you're done for a little bit because you don't have to go into the lagging strand stuff. Ooh, you're not going to make it on this chart. I'm going to have to erase some. Okay, so I'll get through lagging strand and I'll erase and you'll keep going. 3A. So this is going to happen at the same time. And like I said, all of these from this point onwards are HL steps only. So while the leading strand is being built, we're going to start building the lagging strand. And where I said is copied, I'm going to say the lagging strand begins. So the problem is, over here on the lagging strand, we have a primer. That primer is useless to me because of the fact that it ends in 5'. So primer ends in 5'. So the first primer ends in a 5. So that doesn't work. So a second primer is placed closer down. Further from origin. Then we're going to use the same enzyme. So then poly 3 is going to do the same thing, adds the complementary bases. It's going to still do it to the 3' end, and this time it's going to be going backwards. I'm just going to write between the primers. This one's easier, I think, to show it in the little picture. So we'll say adds it between the primers. I'll show you in the picture what I'm meaning by that. The key thing here is instead of copying continuously, it is copying discontinuously. Away from the fork. And then the other key thing is that it's creating these little short fragments, which you definitely need to know the name for, which is Okazaki fragments. Okay, so creating the Okazaki fragments. So this actually involves 2 enzymes because of the fact that I'm placing another primer. I'm going to say that it also uses primase, and then I'm going to keep doing that over and over. So then I'm going to basically have to repeat that. So it's going to use primase, and then it's also going to use DNA poly 3. So same enzymes, but I am going to use both of them. Okay, so again, this time we're really just focusing on the top one. So I can put the bottom one down here, but my emphasis is going to be on the top. There we go. These are not as nicely drawn as they are in my picture, so anyway, sorry. Okay, so again, this was my first primer. And then this primer is 3' to 5' because this strand is 5' to 3'. Woo. As a result, that primer is no good, so it's going to move along, and as it opens up, it's going to place another primer. The 5' end is still no good, but we can copy backwards from this 3'. So it's going to go like that. Then that is going to have to then involve another primer. And then again, I'm going to use that 3' end and go backwards like that. So it's going to keep happening like that. And it is the same enzyme that's doing this. So it is DNA poly that's doing it again. Okay, I'm going to have to erase, but the next 2 points are pretty small. So they're just 2 tiny lines. but we need to then go through the last 2 steps and finish off our lagging strand. So you just keep going, but I have run out of space. So at the end of this, we have these Okazaki fragments. The problem is, hopefully you will recognize in that diagram, is that I have all this RNA stuck in there. So I want to get rid of the RNA that's stuck in there. So now I'm up to step 4. And so what I'm going to do is I'm going to replace the RNA primers. Actually, I'm going to remove that word and say remove. So I'm going to remove the RNA primers and I'm going to fill in with the complementary DNA bases. It doesn't have the word telomeres in this context in the syllabus. It has it later as an example of a non-coding region. So I'm not going to put it here, but remember that the very end primers can't actually be replaced. So they're removed, but you can't actually fill in because of the directionality. And so the ends of chromosomes are telomeres, which are DNA that we don't actually need for any protein, so that it's okay for it to get lost. So that's just a little reminder if you want to remember that. The last primers cannot be replaced. And then I'm just writing the word telomeres. If you're like, oh my gosh, what on earth are you talking about? That makes me sad. Just don't write that. It does come up later as an example of non-coding DNA. So anyway, that's the only context you have to know it, is that the ends of DNA are non-coding. Okay, so we need to take away the RNA and then I want to replace it with DNA. So I need to basically switcheroo the RNA to DNA is my main idea. And the enzyme that does this in prokaryotes is DNA polymerase 1. Most eukaryotes actually use 2 separate enzymes. So we tend to use an exonuclease to remove the RNA and a separate enzyme to do the DNA. So again, this is our model organism to make it somewhat bearable in terms of complexity. So where we left off is we had on our lagging strand, we had all these primers. Like so. And so what we are going to do is poly 1 is going to come along and it's going to remove and fill in. So once we remove it, it's going to try to fill in kind of from the 3' end and fill those in. So the result ends up being that we do get these refilled little ones, but there are gaps between them. It's actually kind of confusing. The gaps are only in one place, so it fills directly from the fragment, but then stops before the next one. Anyway, there are gaps, just believe me. So we replaced my RNA with DNA, but it is still gappy. So my last step in this process is going to be to seal the gaps. So step number 5, and again, we are at HL only because this is a step that is not done, that you guys don't need to worry about because it only relates to the lagging strand. So then the Okazaki fragments are going to be connected. Are connected with phosphodiester bonds. Okay, so now I just need to seal those little gaps, and this enzyme is going to be ligase that does this. And so we started up here, where again I had gaps between my fragments. and then I'm going to come in here and I'm going to just ligase them. So ligase is going to come in, and then ta-da, I end up with one nice continuous strand, and DNA replication is done. The end. All right. So again, if you're SL, all you really need to know is helicase polymerase. If you're HL, you do need to know all of those. The next bit is everybody. So the next part, I'm going to erase this, and then we're going to look at the applications and using what we understand about replication to develop PCR, and then to use kind of that in the context of electrophoresis. Okay, moving on to the applications that have come from understanding DNA replication. So essentially, once we understood the mechanisms of it, we then had the knowledge to then manipulate that and actually do it outside the cell as well. And so that gave rise to this kind of artificial DNA replication, which is polymerase chain reaction. So polymerase chain reaction is just extracellular lab-based replication of DNA. So it's DNA replication outside the cell. So DNA replication in a thermocycler. So a thermocycler is a machine with little metal wells that will actually do DNA replication over and over and over again outside of the living organism to create copies of that DNA. So it's been really valuable for wanting to study DNA, to be able to make lots and lots and lots of copies to facilitate kind of more research, identification tools, and then use in electrophoresis, which we'll cover next. So we'll do a little chart on this. So for our chart, we're going to put step number, just like we did on the last one. For the steps, I want you to know the temperature. Then I want you to know the name of the process. And then we're going to do a little diagram. So on my page, I've only used like half. So half here, and then the other half is for that. So try to make your little table small enough to facilitate still having room for that. So it's basically 3 steps on repeat, and then we'll write at the bottom that you need to repeat that 25 to 30 times. So the first step takes place at a high temperature, so 95 degrees Celsius. So we know that in DNA replication, it's helicase that breaks the hydrogen bonds, but if we just put helicase in there, we just keep breaking them over and over, so it wouldn't work as well. So instead of using an enzyme to make the break, we're going to use heat. So this process is called denaturation, which you should know that term from when heat breaks bonds in proteins. So it's the same concept. So the heat breaks the hydrogen bonds, separating the 2 strands. It's a temperature that's designed to break the weaker hydrogen bonds, but not breaking the actual phosphodiester bonds. So the stronger bonds will be unaffected, but the weaker bonds will break apart. So the heat is going to come along. Here's heat, fire, your love heart, heat. And then what that is going to do is it's going to then create 2 single strands like that. Okay, so we're going to break the hydrogen bonds. Then, like in replication, we then need to actually provide primers to start, because polymerases need primers. Also because a lot of times with PCR, we're not copying the entire genome. We're copying like a gene of interest. So we're going to design primers that like flank the gene of interest and say just copy here. So they kind of have 2 purposes. So it's going to have to cool down a little bit now, because if it's too hot, the new hydrogen bonds won't form for the primers. So we're going to cool it down a little bit. This stage is called annealing, which actually just means to like stick to. And unlike replication, we're going to use DNA primers. There's no reason why to use RNA. It just makes it more complicated because we've separated the strands and they're not still attached. We don't have like a leading and lagging situation. So we can just put the primer on and go at the very end and go backwards, which is what happens. So provided, so we are going to provide these DNA primers, bind to the original strands, and we're going to say bind to the target gene on each strand. So this is kind of the tricky part, is based on what you want to copy. I'm going to actually draw more bases this time to make this point. You're going to have to build primers that fit kind of the goal of what you're trying to copy. So let's say the target gene is, here's our little primers, actually starts here. Then I would put like a little primer here, and if it ends here, I would put a primer there. So that is kind of identifying, this is kind of the area that I'm wanting to copy. So target gene, and so it's saying this is my target gene area. So that's kind of what we're using the primers for, is just to show what gene we're interested in, as well as to get the process started. Then we're going to actually heat it up again. The reason for this is because we've put an enzyme in the mixture that is not going to denature when the temperature goes up. So when the temperature rises, we don't want that enzyme to fall apart and denature. So it needs to be able to sustain the higher temperatures, which normal polymerases would not. But then if we want this process to work quickly, we need to cool it or heat it. It's going to be an in-between temperature. So we're going to heat it, cool it, and then heat it back up or warm it to the ideal temperature of Taq polymerase. So this is my extension phase, and at extension, we're now going to have our polymerase involved. So now we're going to have Taq polymerase, which is from bacteria. So Taq polymerase is a polymerase from bacteria that live in hot environments. They add, again, provided free DNA bases to build complementary strands. So they are going to basically start from the primer. So again, the primer is a really effective way of identifying this is where we want to copy. So here's my primers, like so. And then my polymerase enzyme. So it's a polymerase, a poly3, which is a specific one from that actual bacteria, is going to come along, and it's just going to copy. Like that. So it's going to copy and fill in those kind of gaps. So that's it. So that's all there is to it. Pretty straightforward process. So by the end of this stage, we would have 2 identical strands. So if we want to have millions of identical strands for the applications, we're going to repeat this 25 to 30 times. So this is going to repeat 25 to 30 times inside the cycler. So the cycler is just going to alternate temperatures. So it'll just be like a few minutes like this temperature, a few minutes this one, and so forth, and then just cycle again. And that's going to create millions of copies. And the reason why we want millions of copies is for visibility. So we want to have enough that we can visibly see that. And there's 2 different ways that visibility kind of looks. So that visibility can present in terms of fluorescent tags. So can you be using dyes or fluorescence? And so if you use that, it'll show up. Or the other thing you can do is you can then put it on an electrophoresis gel, and then it will show up as a band on that. So kind of a couple, this is very, anyway, probably a little bit off center. So a couple different kind of ways that can look, but our goal is to get it to the point where it is visible in some way. So we talked a little about dyes or how it's used in things like COVID testing. If we're using it to create identifications, we're going to use it with electrophoresis. You do not actually have to know the step-by-step of how it's done with COVID testing. It's a bit complex because it actually involves reverse transcriptase as well, but that is not named as an application you have to know. So just the general outline of it, I think, is fine in terms of what they could point blank ask you. Okay, so a lot of times that is then paired with gel electrophoresis. So the PCR is making the copies. So this is all about the replicating. The electrophoresis now is actually going to enable us to visualize it. So it's going to actually create a visual DNA profile. And generally speaking, we rely on PCR to do that because, again, for it to be visible, we're going to need enough DNA. So PCR is normally kind of the first step in electrophoresis. Okay, so before we can actually run it through the gel, we do need to cut it. So the way that this is going to work, for it to create like a profile or banding, it has to be different sized fragments. So we're going to take whatever our target gene or gene is. So cut the target gene or genes into smaller fragments. And we actually do that with something called restriction enzymes. It's not named by word that you need that term. I'm going to add it just because I think it's a good thing to know. But basically, there's enzymes that are going to cut the DNA. The key is that these fragments in some way differ by individuals. So the number and size of fragments is different in different people. So depending on what you're doing it for, so if you're doing it to ID someone, you definitely want multiple genes just to be sure. So a lot of these genes might have like 40 different alleles, but that's not, there's other people are going to have the same allele for one. So, but if you use enough of these kind of highly polymorphic alleles, you'll end up with a unique profile. If you're doing it for DNA screening and testing, which is actually not one of the applications you need to know, in that case, you might do just a single gene. So you're just looking at one gene. So you're going to cut it into different fragments, and then we're going to have a gel. So this is the agarose gel, which has pores. So it's a porous gel that the DNA can move through. And then we're going to take our cut DNA and we're going to put it into these little wells. So a lot of times we are going to use the first one might be a known ladder, which is actually something that we already know the sizes of the bands. So those are kind of known sizes. So a lot of times we throw that in, and then we're gonna, after that, put people's DNA that we're interested in. So just to show the example, this would be like, if we're doing it for paternity testing, we'd say, okay, mom has a band here and here. Then we would put in a bands, work out where the child's bands are. So be like, well, the child has a band here and here. And then we're looking to see if that matches. So in this case, what I would be looking at is this band because the child has it, but the mom doesn't. So let's say that the dad shares this band and this band, but not that band. Then we would say, not the dad. So if this is missing, then he is not the dad. But then if we added another potential dad, so this is dad 2, and he actually had a band here and here, then we would say good. So I was trying to draw ones that were on the ladder, not that that particularly matters. Then I would say, yes, he shares that same band, and so I would say he is the dad. So that's kind of looking at how we would actually use it for that. We need to add in a few more details. So we actually load the DNA. So we load the DNA into these wells. So we've loaded this DNA into these wells. But then to make it actually move through the holes, we do need an electrical current. So we then set it up to a current, like so. So this is my current. And the reason it works is because DNA has a negative charge, so it's going to move towards the positive. So negatively charged DNA moves to the positive electrode. So it's going to move down. What's really crucial is that the smaller the fragment, the faster it moves. So smaller fragments move faster. So they're going to move faster, therefore they're going to be further down. So when you stop the electrophoresis, you're going to see it has moved further down the gel in that amount of time. So what we've done is we've created an image that has separated the strands by size. So this separates electrophoresis, separates strands by size. So in terms of kind of what you need to be able to do with these, you would need to have the basic understanding of the relationship between the currents. So the idea that they're moving towards the positive electrodes. So the current is really important. You definitely need to understand this idea that the smaller fragments move further. And this idea that basically we create some sort of identifying information from it. Two different uses that you could need to use this for. So the more complex one is what I just showed you, which is paternity testing. It is named as an application in the syllabus, so you could be specifically asked how it's used in paternity testing. So the key there is that the dad must share bands that the child has, but mom does not. So you have to have the mom's DNA as well to kind of be able to identify that. The other application that it listed in your syllabus is a nice easy one, which is it's used in forensics. So if you're looking for it in forensics, it should be an exact match. So a suspect should be in a match to the crime scene. So these are a little bit easier because you're literally just looking for identical banding patterns. So they're pretty straightforward to do. Okay, so those are your two applications. That is SL and HL, all the same. So there's nothing additional for HL. And I would make sure you know kind of how to explain this, but also how to interpret these. So know it on a conceptual level, but also on kind of a skill level of knowing how to use them as well. All right, coming up next, D1.2. Okay, moving on to protein synthesis now. It's important to note that replication and protein synthesis are totally different processes. Replication's occurring before cell division to replicate the DNA so the cell can divide. Protein synthesis is happening all the time. Every single time our cell's doing anything, it's using protein synthesis to use the instructions to know what to do. So structurally quite similar, but functionally very different. So we're going to do, again, a one-page summary. So the way I have divided it is I've done kind of a picture section and then a step-by-step section. So it's a bit wordy, but they are known for asking you to explain transcription or translation in quite a bit of like stepwise detail. So I wanted to make sure, rather than just having a big picture, that you could also kind of put words to it. So that's how we're going to do it. So we're going to use kind of the first third of the page, maybe a quarter of the page on transcription. Then we're going to use a big chunk on translation, and then just save this little area down here at the bottom for some of the HL material. Another one, the HL, this is all HL. There's kind of some HL embedded into the other. So I'll just kind of indicate as we go. So let's start with approximately slightly more than a quarter of your page as your transcription section. And then go ahead and divide within that section. Do kind of a third or so of it for the step by step. and then the rest is going to be for the drawing. Perfect. Okay, so just kind of dividing the first bit of your page like that. So I'm actually going to start with, I'm going to move this slightly. I'm actually going to start with the drawing and then go back and step by step it. So we're going to kind of overview it, and then from there I'm going to erase this line just because it's not very straight. So we'll just have the line to separate the picture from that. Okay, so I'm going to try to include a lot on the picture. And then, like I said, we'll explain it as we go. So this is going to be step 1, transcription. I'm going to go ahead and outline it as occurring in the nucleus by drawing myself a nuclear membrane. So this is the nucleus. And then I'm just going to put the title down here just because I made some space for that reason. So this is going to be step 1 of protein synthesis, transcription. So a couple of memory clues. Transcription copies. So it's going to copy 1 gene of DNA into mRNA. So if you remember C for copies, the other thing you can think is transcription comes before translation. So C comes before L, so that's another kind of memory trick to remember it. Okay, so up here we are going to have our DNA, which is going to be my original. So there's my DNA. Like so. Might be off the edge a little bit. Okay, and then we're going to try to copy, let's say, 1 gene. I need these, oh, sorry. I know that's annoying when you're trying to draw. I just need a little bit more space before the gene so I can make some promoter space. Okay, so there's my DNA. So if you are HL, you need two little things to add on. So when I want to start transcribing, kind of ahead of the actual gene is going to be a promoter region. So this is my promoter. Often the promoter ends in a TATA box, TATA box, which is actually TATAAT. It's just a bunch of A's and T's because that makes it easier for polymerase to make cuts because there's only 2 hydrogen bonds. So it's like land here and then start cutting at the TATA. So that's usually kind of where the cutting starts, is at the end of the promoter, which has a TATA box. Most of the time, it's actually not polymerase that binds directly to the promoter. Most of the time it's a regulatory protein called a transcription factor. And that is essentially just to ensure that you can turn genes on and off. So rather than them being transcribed all the time, they're actually only transcribed in the event that the transcription factor is there. So it's just a cool kind of regulatory mechanism. And so then my RNA polymerase is going to bind there. If you are HL, you need to know all of that. If you're SL, you don't need to actually know the terms promoter and transcription factor. You can just know RNA polymerase just attaches on. All good. So now RNA polymerase is going to move through. Let's label our strands. So this down here is going to be oriented as the 3' to 5' strand. This up here would be the 5' to 3'. That is important because we don't want to transcribe a lagging strand. I only need a single copy, so it's going to be a lot more sensible if my single copy runs 3' to 5'. So that's going to be my template strand. Up here, this would be my sense strand, but I don't copy my sense strand. So the template strand is the one that is going to be copied across from, and it should be read 3' to 5'. So DNA is 3D, so it needs to be oriented so that the strand you're wanting is running upside down. So it needs to be oriented in that direction. Let's add some bases. T, A, C, G, A, that's T, A, C, G, T, A, etc. And so then RNA poly is going to come along and it is going to move along the strand. And then as it moves along, it is going to be building the complement. So instead of T, it's going to be A. Instead of A, it is U. So make a note that when we are doing transcription, we are using uracil. And then as it kind of moves along, it'll keep building. So it'll be C, A, U, G, C, and so forth. So it's going to copy that messenger RNA from the template strand. So it's messaging the complementary sequences to the template strand. Then, after it's done, it has created mRNA that is made. This mRNA is not ready, though, to go out and do all it needs to do. Two reasons. First of all, within it, it has these introns still. So introns are non-coding areas. So they are in the middle of the gene, but they don't actually code for a protein. The non-intron areas are exons. So this is going to be exon, exon, exon. So the non-coding is the intron, and the coding area is the exon. So before it actually leaves, some modifications need to be made to it. So we need to make some post-transcriptional modifications. Now these post-transcriptional modifications, first of all, only occur in eukaryotes. And second of all, less important in the overall scheme of biology, but they are not important for you if you're SL. So these are HL concerns only. So SL, you don't need to worry about those. Okay, so when that happens, what they're going to actually do is they're actually going to, ends up being the third thing, but you don't need the sequence. So we're going to remove all of the actual introns. And so that's going to be done, let's move it a little bit lower, with something called a spliceosome. So this is going to be exon only. So these are going to be cut out using a spliceosome. And it's going to come along and remove these. And then it is worth remembering that the reordering of the exons can be changed. So exons assembled and the order can be changed. And if you change the order of that, that's called alternate splicing. And alternate splicing is just a way of making one actual gene produce different proteins. So basing on how you change the actual order, you can use a single gene and make more things. So it's like taking one single recipe and changing the recipe and being able to make different cupcakes, muffins, cookies, whatever, off of a core recipe versus needing a different recipe for every single different variation. Then on top of that, we need to add some protection because the cytoplasm is a scary, scary place. And that's because of things like having the proteasomes and proteases there that are kind of intended to break things down. And there's nucleases. So there's enzymes that have kind of cutting power. And that's intentional so that they have the ability to cut apart any sort of mRNAs that we don't need anymore. But if we want the mRNA to not get cut up, we need to give it some protection. So we are going to give it some protection in the form of down here on the 5' end, a GTP cap, and that is going to provide energy. So it's actually 3 phosphates, and it's also going to provide some protection as well because the GTP cap is actually methylated, and so as a result of the methyl, nucleases can't actually cut from it. So it ends up blocking the nucleases, the enzymes that try and break down the mRNA. Down here on this end, we have a whole bunch of A's, like so. So that is going to be my poly A tail. And that poly A tail is for protection. So essentially we know that enzymes in the cytoplasm will actually degrade mRNA. And so by putting a nice long tail on, it gives it time. So it slows down the degradation of mRNA so that it can be translated a few times. If a protein and an mRNA is for a protein we need lots of, it might have a really, really long poly A tail, so it takes a longer time for it to be degraded. So that's protecting it from being degraded or broken down inside the cytoplasm. Then once all of that is done, then this can actually leave the nucleus. So then it's going to come out of the nucleus and travel along on its own little way until it reaches a ribosome. So this is going to exit the nuclear pore. And so the mRNA exits through a nuclear pore. Okay, so that is transcription in pictures. What I want to do is just kind of summarize it in worded form as well. If you feel like, nah, I'm really good on the picture, then you can always leave this part off. But I just wanted to make sure if you were to have to actually write about it, because it is a really commonly asked kind of ERQ, that you could write about it in words as well. So I'm gonna basically look at it as kind of a 4-step process. So the first step is gonna be HL only, which is kind of looking at the transcription initiation of it, although part of it you kind of need as well in SL. You just don't need the word transcription factors or promoters. So we are going to say that a transcription factor, I'm just trying to use the colors that I used in the picture, is going to bind to the promoter and then once the transcription factor has bound to the promoter, that is going to allow RNA polymerase to bind. And RNA polymerase is going to bind there, and it's going to start cutting to open up that gene. Stop doing that. To bind, and cuts at the ta-ta box to open up the gene. Okay, so that's kind of how does it get started. Those are the steps involved in getting started. Then let's look at what is actually happening at the transcription level. So then once that is open, RNA polymerase is going to move along the gene. And you are going to note that it is specifically moving from the 3' end. So it's going from the 3' end to the 5' end of the original. It's adding complementary RNA strands, or RNA bases. And the outcome of that is to make a 5' to 3' mRNA strand. Okay, if we are looking at, for HL, we need to also then talk about the modifications that need to be made. So after it's built, we need to do post-transcriptional modifications. And these are still happening in the nucleus. And so my post-transcriptional modifications that we're going to make, it's actually 3 things. So the GTP cap actually comes first. So GTP cap at the 5' end. I'm not going to write energy again because I already wrote it there. Then we're going to add a poly A tail at the 3' end. and then the introns are going to be spliced out. So then my spliceosome removes the introns. Okay, so once all of that is done, the last thing that is going to happen is the now mature mRNA is going to leave the nucleus. So if you were writing about transcription, you could write about it in basically 4 steps and go through that. Again, if you are HSL, you don't need the term transcription factor or promoter, although you should still discuss RNA polymerase landing and making the cut. And then you don't need the post-transcriptional modifications. So you could omit that part of talking about it at an SL level. Okay, so now I'm going to keep going for our next section. So the translation is probably a little bit bigger, largely just because there's more steps you need to know. So translation's at a pretty high level. There's an initiation part that SLs don't need to know, although I think it makes good sense. So I wouldn't worry about the terminology, but I don't think it hurts to understand it in order to make sense of the rest. But technically you can go straight to kind of once it's already underway. But then the actual steps of it you are accountable for at SL as well. So make sure you have a pretty good understanding of this because it's probably the most complicated of the things that you're learning at an SL level. All right, so again, I'm going to make a space and I'm going to kind of come back and do some steps to go through what's actually happening to show that. So I'm actually going to start with the initiation steps before I even do my diagram, only because at my diagram I want to like show it all underway. So our mRNA is now arriving somewhere. So don't draw this, I'm gonna draw it, but before the mRNA can actually bind to it, the ribosome is actually already getting ready. So at the initiation, what is actually gonna happen is what's called an initiator tRNA. So we're gonna say initiator tRNA. And what makes it an initiator tRNA is it carries the enzyme, the amino acid methionine. So with a methionine amino acid and a UAC, yes, I was checking my head, anticodon. So that's kind of the key. So for it to be the initiator one, it needs to actually have the anticodon that matches the start codon. So that's kind of the key feature there. So that is going to be my initiator tRNA. And it has the first amino acid that's going to be added. And its anticodon is a match to the start codon. So that tRNA is actually going to bind to the small subunit of the ribosome. So let's go ahead and draw a ribosome. I'll draw it later because it hasn't actually attached yet. So that is going to bind to the small ribosomal subunit. Then those two things together, which you can see called the ternary complex, you don't actually need that terminology. It's like not in your syllabus. So I wouldn't worry about that, but I'm just going to use it because otherwise it's actually quicker than writing initiator tRNA plus small ribosomal subunit. So if you can remember the term, it saves you writing that, but it's fine. You can just say what it is otherwise. Bind to the small ribosomal subunit. Oh, sorry, bind, that's already part of the small ribosomal subunit. Bind to the messenger RNA. Okay, then they're actually going to bind at the 5' end. So they're going to come right down here, and bind at the 5' end. Like so. Okay? So that's going to happen. Then they're going to keep moving along till somewhere along here they run into AUG. So AUG is the start codon. So the process does not happen until it hits the start codon. So once it hits the first start codon, then the large subunit is going to join, and they're going to kind of join together. So it's going to join there, moves towards the 3' end until it hits first AUG. So once it hits the first AUG, it has found the start codon. At that point, the large ribosomal subunit joins. And the initiator tRNA at the P site. Okay, so we have to break a few things down. So we'll get to our ribosomes now. So I'm going to call this initiator tRNA, tRNA1 on my picture. So that's going to end up being tRNA1. So that's going to be tRNA1. Okay, so that's going to end up at my P site. So on my drawing, I'm going to actually have it already kind of exiting. And let's go ahead and get our ribosomes joined together. So that's going to be the small subunit. And of course, this is going to be my large subunit of the ribosome, but I've already written it over there, so I'll leave that off here. Okay, on my ribosome, there is a kind of space equivalent to 3 codons. Those are called, the areas there, the APE sites. So those are the 3 sites of the ribosome. You don't have to actually name them APE if you're standard level, but you do have to explain what's happening at each one. So to be honest, I don't really think it's that much more complicated just to name them because you have to explain them anyway. Okay, so remember these on the mRNA, these are the codons. So that is a codon of mRNA. Okay, so now my first tRNA is sitting at my P site, and then the next tRNA is going to come along. Perfect. So the next tRNA, and so this guy we're going to name number 2. And we're going to draw it at that. I'll show you why in a second. So the next tRNA is going to arrive at the ribosome. It is going to arrive at the A site. And it is going to be carrying the amino acid for the next codon. And it is going to have an anticodon that makes sure it gets into the correct spot. And it has the complementary anticodon. So everything kind of going from here is what you would definitely have to be able to talk about at SL as well. So you don't necessarily have to focus on what gets the ribosome joined together, but you do need to focus on this part. As soon as that arrives at A peptide bond forms. So as soon as tRNA2 arrives, so once tRNA2 has arrived, the A site, a peptide bond forms. And the methionine amino acid is transferred to that tRNA. So as soon as it happens, that is what's done. So this is my next tRNA. So this is tRNA 2. It is arriving at the A site, like so. As soon as that happens, it's going to form a peptide bond. That methionine is going to get moved over to that tRNA. So that methionine is moved over to tRNA 2. As soon as tRNA 2 now has the protein chain, it transfers over to the P site. So then the ribosome moves. So tRNA 2 is in the P site, holding the protein. Okay, so this is where I want to draw kind of where we are at right now. So this is saying, here is tRNA 2. We're going to show it in the P site. So we're going to show it's already moved over, just because that's the one we're kind of focusing on. CCC codes for proline. Its anticodon would have to be GGG, and we'll give it a proline amino acid. We'll talk about the codon chart in just a second, so we need to come back to that. So remember, what happened was this moved over because a peptide bond formed with that, and the methionine that was on tRNA 1. As soon as the methionine is gone and this has moved, so the tRNA 2 is now in the P site, the original tRNA 1 moves to the E site, and leaves the ribosome. So the first one, once it's no longer holding the chain, it is going to be leaving. So it used to have met on it, but now it doesn't need to stay anymore. So it's like, see you later. So it is currently exiting from the E site. And of course, it would have UAC. So that was my initiator tRNA. So that's tRNA number 1. This is tRNA number 2. And it's currently, I'm showing it at the P site. So this is why I had to do the steps, because it's really impossible to draw this happening in time because it's kind of changing all the time. So it's a little bit trickier. So this is where we're at right now in my steps. So right now in my steps, we can see this one had already arrived previously. It caused it to start. It was in the P site, but as soon as 2 arrived, it moved it over. The same thing will happen now. So once the new tRNA 3 arrives at A, the new bond will form, and ribosome shifts down. So, while this has happened here, it's already arriving that the next tRNA is coming into play. So here's number 3 arriving at the scene. He is going to have the amino acid that is complementary to UUU, which is phenylalanine. So he's going to have Phe, and he needs to make sure it lands in the right place. So it's going to have AAA as its anticodon. And then again, we can't really show it happening right now, but once that lands, boom, proline and phenylalanine will join together, and then everything will shift. It's actually the ribosome that moves, not the tRNAs. So the ribosome will move down, and as the ribosome moves down, they move over a site. And then this will just keep happening. And so this will now repeat until a stop codon is reached. So again, if you are talking about it at an HL level, you will need to actually identify them as P sites, E sites, A sites. If you are talking about an SL level, you don't actually have to refer to the site. You can just basically say the next amino acid comes in, the bond is formed. So you still have to know kind of all of it except for these 2 steps. So step 1 and 2 are HL only, but basically everything else is SL as well, but you could get away without using the name of the sites. So a little bit of flexibility there. Okay, let's add a couple more things to our picture. So first of all, let's label these sites now that we've talked about them. So this site here where it is arriving is the arrival site, which is my A site. So this is A for arrival. The site where the actual protein is being held is the peptidyl site. So that is going to be my P site. And then the site where the tRNA exits from is going to be my exit site or my E site. So even though it looks like this, remember the order is actually that tRNAs go 8. So they arrive at the arrival site, they move down to the peptidyl site, and end up exiting at the exit site. At any given time, only 2 RNAs can actually be at the ribosome. So the one that's at the P site and the one that's coming in at the A site. The E guy's already leaving. So you can have 2 bound at the same time. That's how the peptide bond forms. But as soon as it forms, the shift occurs. So basically the A and P sites should kind of be filled and the E site should be exiting so that as soon as it forms, it can move along. Okay, a few things we want to add kind of just to clarify things. So here's my codon, here's my anticodon. And we should note that the codon and the anticodon are complementary. So these 2 are complementary, and that ensures the correct placement of the amino acid. So the anticodon is basically like a little address tag. So let's talk for a second about the actual amino acid. So what determines the amino acid? So the amino acid for the next codon. And we can say here determined by the genetic code. And then let's kind of look at what we mean by that for a second. So it is always the mRNA codon that actually determines the amino acid. So the mRNA codon determines, let me make sure it's still visible, determines the amino acid. Also, I realized we haven't actually written our step, because you know how here we wrote like step 1. So let's go ahead and add over here that we are now looking at step 2. And that step 2 is translation. And in translation, we are moving from mRNA to a polypeptide. Okay, so we said the mRNA codon determines the amino acid, and it does that by the genetic code. So the genetic code is the specific instructions as to which codons determine which amino acids. Another way, the genetic code is the official term for it. When we try to represent it ourselves, it's represented as a codon chart. So when we say genetic code, that is the same thing as the codon chart. So the codon chart is our visualization of that. So again, a reminder is that when you are using the codon chart, you always use mRNA. So anytime you are looking up anything on the codon chart, make sure that you are looking up mRNA. There's two other vocab terms you need for the genetic code. So the genetic code is universal. So it is the same in all living organisms. So in every living organism, U would always be phenylalanine. And the other term for it is that it is degenerate. And that just refers to the fact that there are 64 codons, but only 20 amino acids. So there's going to be overlap. And that's significant because that's why we have silent mutations. So that'll come up in our mutations part. Make sure everybody knows how to use this. We did quite a lot of practice in class, so you should be pretty good on that, but make sure you know how to use the codon chart for your test, for your exam. So definitely want to make sure skill-wise you're up to date with that. The last little thing I'll just draw over here is that while all of this is going on, we are already preparing the next one. So this is an enzyme. It's an anabolic enzyme. It has a name, but you don't need to know it. It's aminoacyl tRNA synthetase. And so while this is all happening, the next one is going ahead and adding the correct amino acids onto it so that it can kind of keep moving along. So this is kind of going on in the cytoplasm where it's basically attaching that onto it. So that's kind of preparing the next one along for whatever it is going to be. So that would be kind of getting ready for the next movement. So before that kind of is ready, that's kind of combining the amino acids and the tRNA in the cytoplasm. So it's kind of attaching amino acids to tRNA. Okay, so that's the process. Again, I wanted the steps because in some respects, this is one area where the picture might be slightly less helpful because you just can't show the sequence very well. So this is like a freeze frame that at least shows what all we're talking about, but make sure you could put it kind of step by step and spread it, you know, and account for it all happening in terms of written steps as well, and make sure you could explain it. It comes up quite a bit in extended response questions, so it's definitely something to know. For SL, that is all for this little subtopic, so fast forward a little bit to get to the mutation stuff. For HL, we have one little last component, which is to look a little bit at some of the modifications and things that happen after translation. So it's kind of two little HL only subparts that are not super related. So modification of proteins and recycling of proteins. These are both fairly small points, so we won't spend ages on them. So modification and then recycling of proteins. Okay, and again, both of these are going to be HL only points. So we're just going to indicate that we're only talking about higher level in this part. Okay, so essentially when we talk about modification, and this hits well with our protein structure stuff, that we're basically saying at the end of translation, we have a polypeptide, which is just a chain of amino acids. So it really only has that primary structure. And then we need it to turn into a protein. So this is looking at the steps that are needed to turn the polypeptide into a protein. A lot of times this happens in both the endoplasmic reticulum and in the Golgi apparatus. So this happens after it's left the ribosome. And you can think about this as things like tertiary folding. It's things like quaternary assembly, if there's multiple parts. These are the kind of things that have to take place to go from a polypeptide to a protein. There is a named example in the syllabus, so we're just going to go through that named example because it also has examples of the kind of things that can happen. So the example you need to know is the steps that go between pre-proinsulin to proinsulin to insulin. Like so. Okay, so those are going to kind of be my 3 steps that are going to be involved. And then you just need to know kind of what happens in each of these stages to go from that as kind of examples of what modifications can be. So for instance, the first thing that happens is the removal of the first few amino acids, including methionine. So a lot of times MET, or the first couple of amino acids, are removed. So every protein starts with MET, but MET doesn't necessarily structurally make sense in every protein. So a lot of times that's removed. The next thing that happens is the disulfide bridges are formed, which is going to be important. And then once those disulfide bridges are formed, we have proinsulin. And then the last stage is actually that it is cut. So actually breaks bonds to remove a section of amino acids in the middle. You don't need to know how many. I'm having to look it up. It's 33. So I don't know how many, but a section of amino acids. And so by the end, what it ends up with is basically an alpha chain, a beta chain, and they're attached by disulfide bridges. So it ends up creating, that's a terrible little tiny drawing that does not work at all. I don't think you actually need to know it, but you basically have an alpha chain, a beta chain, and they are actually bonded by these disulfide bridges. So that's what in the end insulin ends up looking like. And so you actually cut out the DNA that connected them initially. So they end up being 2 detached strands that are attached by disulfide bridges, but they actually came from one giant chain of amino acids. So I don't necessarily wouldn't worry about that a ton, but you could need to be asked specifically about that, or just generally talk about the removal of methionine, the addition of R group bonds such as disulfide bridges, the potential for removal of parts of things. Okay, last is recycling of proteins. So basically, at times there are proteins that have lost their function, and so they are either denatured, they are no longer needed, so their function is fine. It's just the cell no longer needs it for where it's at in its kind of life cycle. Or it was damaged from a mutation, so it was a faulty protein. But if we have a large protein needing to break down, instead of using small proteases that can only really break single peptide bonds, we use this massive thing called a proteasome. So this is the proteasome. And it makes this kind of cylinder-like shape that is full of catalytic enzyme activity. So it's basically a large protein that has kind of multiple catalytic sites. So rather than breaking it one at a time, it can enter and kind of get broken from there. So here's my SAD protein. So this is my non-functional protein. And so it actually enters this little core. And the way that it knew it was supposed to break it down was because attached to it was this chain of ubiquitin, which is ultimately going to be broken down as well. So this is going to be my tag, ubiquitin tag, that basically says it needs to be killed. I call it ubiquitin because ubiquitin being a protein. So it's actually pronounced ubiquitin or ubiquitin, but you can call it whatever because it is not a verbal exam. So ubiquitin being a protein if you have a UB tag on you. So it identifies that tag. It goes into the cylinder. It will end up recycling the actual tag itself. And then it will also break down the protein into amino acids or dipeptides. So sometimes they'll break it into dipeptides that you can then use proteases to further break down. And so the protein parts are recycled. So this way we can reuse those amino acids for other good purposes in the body. And similarly, we will reuse the ubiquitin so it can tag another one. So again, all you need to know there is just the idea that we can break down proteins that aren't functioning and recycle their parts. I would know the terms ubiquitin and I would know proteasome, and that's basically it. All right, last part will be on mutations, D1. Okay, last section for D1 on mutation and gene editing. The mutation stuff is identical for SL and HL, and then the gene editing part will be HL only. It's just going to be the very bottom part. So HLs just try to leave a quarter of your page to hit the editing components at the end. SLs, you can take a little bit more space. All right, so like I said, everything for mutation is the same, so that's just the editing part that we'll add. So what I want to try to organize this is as we look into mutations, how do we understand what the impact will be? So as we go through the impact, we'll hit kind of our terminology. So when we talk about this, we're going to say the impact of a mutation depends on several different things. So we'll define mutation as we go, although hopefully we already kind of know this. But as we kind of look through what we're looking at, we'll kind of go through it bit by bit. So when we talk about a mutation, we are talking about any random change in the genetic material of an organism. So any change to any sort of genetic material. I use that slightly broad term because it is a change to DNA, but sometimes it removes like whole chromosomes. So changes like that would still be to the genetic material, so we would still classify it as that. So the impact depends on a bunch of different factors. So we're gonna kind of go from big to small. So the first thing that it can somewhat depend on is the cause of the mutation. So different things can cause mutations and they tend to have slightly different impacts. So you need to know two different main categories of things that cause mutations. So there's essentially kind of an external versus an internal cause. So one possibility is that the mutation is actually caused by external exposure to things called mutagens. So a mutagen is a external thing that causes mutation. And these mutations, these mutagens, tend to break DNA and cause fairly dramatic impacts. So external mutagens that break DNA and lead to kind of poor replacements. So they tend to break DNA, similar to how a gene knockout works, that break DNA and cause kind of mutations in the replacement. So most of the time, if a mutation is due to a mutagen, it tends to be worse. So it tends to be more likely to affect several different bases or several different areas in the body at the same time. So mutagen-based mutations tend to be a little bit more significant. Within mutagens, you do need to know two different categories. They can be chemical. Lots of different examples of that. We'll use benzene as an example. Benzene is a chemical that's found in polystyrene and nylon. So plenty of fairly innocuous products do have that chemical in them. And then the other category for these type of mutations is radiation-based. And the best example that we're all exposed to all the time for that is UV radiation. So ultraviolet radiation causes mutations as well. So again, mutations that are due to a mutagen tend to have a little bit more significance in terms of making more changes. The other cause of mutation is just random errors during DNA replication. And this happens as well. These generally are pretty rare because of polymerase editing. But every now and then a base is simply put in incorrectly or left off or something like that. And these can cause mutations. So generally these cause ones are smaller scale, mutagens are larger scale. But we will make a note that this is rare due to poly 3 proofreading. So most of the time these do not happen. And then mutagens are also more rare in the sense that they can be avoided, even by things like applying sunscreen and so forth. So that's kind of the first thing when we talk about mutations is, well, what caused the mutation? Because that might change its impact. The next thing we need to look at is what type of cell did it happen in? So again, there's kind of two broad categories here that are going to have really different impacts in terms of what kind of cell they happen in. Most of the time, these mutations happen in a somatic cell because we have a whole lot more somatic cells, and we make somatic cells a whole lot more often. So just statistically, this is where most of them are going to happen. So they are happening in a somatic, which is just another name for a body cell. And so what happens is when that body cell actually divides and replicates, it can cause a cluster of abnormal cell. So it can lead to tumor formation. So it plays a role, therefore, in cancer. So it is still significant. So we don't want to make it sound like these are not a big deal because they do lead to tumors. However, they are not passed on. So they do not get passed on to other generations. So they only are going to impact that individual. So they won't get passed on to their offspring, so it's kind of a one-off mutation that impacts the person. The other option, and this can happen via the random errors in replication during meiosis. It can also just happen due to exposure. So gametes can be damaged just due to exposure to mutagens, but it happens in a gamete, which is a sex cell. So if this actually is fertilized, so it's only important to fertilize, then every cell in the individual has the mutation. And this is going to lead to genetic disorders forming, and they are passed on to offspring. So then the next generation can end up with the mutation as well. So those are going to have a bigger impact, big scale because of that. So it can lead to genetic disorders and it will be passed on. Do want to note that not only can it lead to genetic disorders if it's negative, but if it's positive, it can actually cause natural selection and be positive. So we'll make a little note of that here as well. If it's a positive change, it can then activate natural selection. But again, that is only the case if it was in a gamete to begin with. So if it was in a gamete, then it has the potential to be passed on and has the potential to prompt natural selection. We'll look at an example of that in a minute. Next thing we need to consider is essentially how much was changed. So within that cell, what was the degree of change? So it could potentially impact the entire, a large part of a chromosome. So could it be a chromosomal mutation? Could it be a whole area, a large section of DNA, which might be a whole chromosome, it might not be big enough to be considered a full chromosome, or one single coding region? And of course, it could happen in a non-coding region as well. So the other possibility is it's a mutation in a small area, but it's simply non-coding. So if that, of course, happens, it's not going to have as big of a deal. So if that is the case, we call these gene mutations, and that's what we're talking about for this unit, because these are the ones that, of course, are going to impact proteins and relate to protein synthesis. So mutations that happen in a single coding area are called gene mutations, and that's going to be our emphasis. But we do want to be aware of that kind of idea that there are other forms of mutations out there. The next thing we would want to say, well, it also depends on the protein's function. So essentially, if the protein has a vital function, then mutations are going to have a much bigger impact. So the impact depends on the significance of the protein. And I'm going to do a little HL thing right here, just because it makes reasonable sense as to where to put it. So basically, if a protein has a vital function and a mutation occurs in it, the mutations are often like life-threatening, and so they actually will get eradicated by natural selection rather than staying in the population. And so as a result, really vital proteins tend to not accumulate mutations over many generations because they tend to get eradicated, leading to highly conserved sequences. So mutations in vital proteins are usually eradicated. So I'm just meaning like gotten rid of, killed off, if you will, by natural selection. So these genes end up not having a lot of differences between them. So the result of that is that they generally result in these conserved and highly conserved sequences. This is HL only. So sorry, I said the mutations was the same. I am throwing this one little HL point in there. So this is just a terminology that you need. So they result in conserved sequences. So they're conserved sequences, meaning that in general, like the protein for CFT, which relates to transport, that if damage causes cystic fibrosis, the vast majority of the population, 99%, have pretty much the exact same strand for that, the same sequence, because it's a really important protein. So that basically means that they are identical sequences within a species or similar species. So even they found that like that protein is quite similar in mice as well, because they have a similar kind of thing where they have to transport salts in order to maintain osmotic pressures and so forth. Can also, if it's really important, result in highly conserved sequences. And highly conserved sequences are identical over long periods of evolution. And these tend to be the same in lots of species. So these are really important metabolic proteins like for helicase, for things that are related to aerobic cellular respiration like cytochrome C and things like that. And so most why these exist is due to this. So these exist due to functional requirements. So basically they're really important for the organisms to function. They can also be due to low mutations rates. So some areas of the genome simply don't mutate as much, particularly if they're in kind of highly coiled areas. So sometimes you will see conserved sequences in areas that are not functionally important, and that might just because they have the low mutation rate, but most of the time these conserved sequences are the result of a functional importance. Okay, so that's everything down to the type of mutation. So I'm going to erase that. So the last thing that determines how serious the impact of a mutation is the actual type of mutation. So we're going to show this in a little chart. So we've kind of worked all these things matter, but at the end of the day, the last thing that says how much does this mess you up is the type of mutation. So we're going to make a chart for this one. So we're going to put type of mutation, and then for that type of mutation, what I want you to know, I don't know why I wrote that in. We're going to say for that mutation, what happened to the DNA? We're going to say what happened to the amino acids. Then we're going to say what does that do to the protein? And then if relevant, what is an example? Okay, so that's going to go kind of all the way across your page. So there's 5 that you need to know about. Again, it hasn't actually specifically used the words missense, nonsense, and silent, but it does say you have to know that sometimes it doesn't impact the protein, and then it does also mention the like examples that are nonsense. So we're going to go ahead and learn the terminology because I think it's a nice thing to use to kind of clarify what you're talking about. The term you do definitely need to know is substitution. And then within substitution, we are going to do the 3 types. So substitution missense. So in any substitution, the impact to the change of the DNA is one base is changed to another base. So a single base is changed. If it is a missense mutation, the outcome of that is one codon changes one amino acid. So one changed codon changes one amino acid. In terms of what does that do to the protein, usually the protein is still produced. So the protein is made, but it has a different structure. And how different the structure is depends on kind of how similar the R groups were. So it has some sort of structural change, and we'll just put R groups dependent. Sorry, that's not super clear, but I'm trying not to write a ton of words. So it depends how similar the R groups are. And if you change the structure, generally you will then have a functional change or a functional loss. There is a possibility for that functional change to be positive. For example, you need to know an example. It doesn't say you have to know which one, but we'll go ahead and learn sickle cell anemia as our example of that. So sickle cell anemia is an example of a missense mutation. All right, next one would be a substitution nonsense. So in this case, the actual change to the DNA is the same. So there's no difference here. So same as above, or you can just draw an arrow. What is the change to the amino acid? So this time, what was previously a codon is changed to a stop codon. So no more amino acids are added. So the result is you end up with a shorter protein. And depending on where the stop is, so if it was close to the end, it may maintain some sort of function, but usually it is non-functional. In fact, usually it would just be recycled by a proteasome. So most of the time this will create an entirely non-functional protein. And you don't necessarily need, there's lots of things. So some forms of hemophilia is an example, but I wouldn't think you need to specifically worry about how, but just out of interest, one of the most common mutations that gives rise to hemophilia is due to a nonsense mutation. All right, moving along. The third thing that can happen that is still a substitution is a silent mutation. And again, this is going to be same as above. But this time, it still changes the codon, but there is no change to the amino acid. So the changed codon codes for the same amino acid. And that's, again, because the genetic code is deleterious, and so it is possible for multiple codons to code for the same amino acid. It is going to have no impact on the protein, and as a result, there are no examples of it because it does not relate in any sort of disorder. So we would never see any examples because it does not cause disease. It also does not do anything helpful. So it does not cause disease, it does not lead to genetic variation, it basically does nothing. Okay, so those are my three that are substitutions. Then we need our, done. Then we need our last two, which are insertion and deletion. So insertion. So this time one or more base is added. What that does to the amino acids depends on if it creates a frame shift. Usually it does. So when it causes a frame shift, it basically is exactly what it sounds like, meaning every codon after is changed. So that is going to change every amino acid and therefore, or all codons, therefore amino acids after the mutation. The only exception to that is if it's a multiple of 3. So it's 3, 6, 9, any of those, then it wouldn't shift everything. It would still have an impact, but it would not have as much of an impact, which actually is what our example is. So usually it causes frame shifts, but not always. Impact on the protein is frame shifts usually create a non-functional protein. If it is not a frame shift and it is just an addition of a base, it might be a little bit different. So in this case, the example that you do need to know is the 5-HTT gene, which is related to Huntington's. and it is actually a trinucleotide repeat. So it just keeps repeating 3 nucleotides over and over and over. And so in this case, the protein is still functional, but has structurally compromised. And depending on how many extra repeats there are, that impacts whether it's a complete loss of function, kind of the severity of it. So you just need to know that Huntington's is an example of an insertion mutation. And then lucky last, our last one is a deletion. So with a deletion, a base is removed. And it can similarly be multiple bases. Again, also usually causes frame shifts. Not always, but usually. Which again is going to change all the amino acids after it. Again, another example, cystic fibrosis is actually an exactly 3 base deletion, so it does not cause a frame shift. So not always, it has to be a not multiple of 3. Again, usually protein loses function is usually the outcome. That is not always negative. So here's our cool example they want you to know is the CCR5 gene. And when it is non-functional, that actually provides immunity for HIV. So the non-functional version of that gene can actually provide protection from that actually entering into the cells. So not having the mutation could actually be good. And when you have a positive mutation, it could increase by natural selection. So it hasn't really happened yet because HIV is too recent. But when you have a mutation like that, it can provide a survival advantage. Individuals that have it therefore out-survive those that don't, and it could increase in some areas that are heavily affected by HIV. It could actually increase in frequency. Okay, so that is all things SL. HL will just do about 10 more minutes and just pick up the biotech stuff that we need to finish up with. All right. So for the biotech component, there's really just 2 things. We already covered the conserved and highly conserved, which didn't really fit into biotech, but that was the one other little bit of HL only. So for the biotech component, we have gene knockouts and then CRISPR. They are somewhat related because the gene knockouts are often now made via CRISPR. So we'll go into these first. So when we say gene knockouts, we want to make sure we know what do we mean by a gene knockout. So a gene knockout is when a single gene is deactivated. It could be deactivated, or it could be removed, or it could be mutated to loss of function. So something like that. So there's some sort of severe impact to it, and then loss of function is observed. So the purpose of these is to use that information to determine what the function of the gene is. So it basically is a test for gene function. So if the gene is gone, what happens as a result is the purpose of the knockouts. They are done almost always in model organisms because these kinds of processes are not able to be done in humans. So model organisms are needed because they're going to be ideally organisms that have many short generations. Because these have already been made, there already exist libraries of them. So there are libraries of knockout organisms available. By available, that does not just mean you could go be like, hey, I want to hang out with some knockout mice this weekend, but it means if you're a researcher who's like, I am researching the impacts of hemophilia, they'd be like, great, we already have mice that have been bred with many generations that have that gene knocked out, so you could actually access them because they're quite difficult to make. So now that they've been made, there's continuing to be kind of generations continued of these. And then now, these are often made by CRISPR. So historically, they had to be done other ways, involving removing embryonic cells and trying to reinsert them, but they are now done using CRISPR. So that is a good segue into what do you need to know about CRISPR. This is just a reminder that this whole section is HL only. All right, CRISPR-Cas9. And I always say to my students, they should never be abbreviated to CRISPR. It should always be abbreviated to Cas9 because CRISPR really has nothing to do with it anymore. So CRISPR is kind of how we discovered it, but Cas9 is the real hero. So this is our gene editing tool. Okay, so the actual CRISPR part is kind of a bacterial immune tool, and this is used to build guide RNA in bacteria. You do not really have to know about CRISPR, but that's what the guide mRNAs are made from in bacteria. And of course, we are just going to go straight to making them. So CRISPR is where the guide mRNA, our guide RNA, comes from. Cas9 is the actual DNA cutting enzyme that does the actual editing. So Cas9 is the star because it's going to actually make the break. The CRISPR is just gave our inspiration. So essentially what we need to do is we are going to actually make our own guide RNA. So this can be made in a lab. So it's normally made in bacteria, but we have worked out how to kind of replicate that. So what happens is you identify a strand, build a strand that is complementary to the target DNA. And then it's kind of attached to a loop that is actually what enables it to attach onto Cas9. So this is my palindromic repeat. In some cases, you might also add a template DNA. And so you can add a repair template. So it's possible that you could go in and also kind of add a little section of what you want to repair. So that can be done if you're actually editing. If you're just making knockouts, you don't need to do that. So that is my guide RNA. And then it needs to be attached to my Cas9. And my Cas9 has cutting abilities. So it has some DNA cutting sites. So then when you put those together, that basically enters into the organism. And so floating throughout the organism, it would encounter the target gene. Somewhere on my target gene, there would be a AGG, some sort of GGG, a PAM sequence. What would happen is my Cas9 would come and land on the CAN sequence, and then it would only have identified it because of the complementary guide. And then This is how it, of course, attaches onto it. And then it would come along and it would go chop and it would cut the target gene. Once it's cut the target gene, there's basically two possible outcomes, is that you can just allow it to repair automatically. And if you allow for it to self-repair, it's probably going to do a bad job of that. And that's going to cause mutations. And that's going to create a knockout. So because it doesn't specifically say kind of how you have to use it, this is probably the easiest thing to explain, is you just cut the DNA. When the organism tries to repair it, it usually makes like an uneven cut. So it cuts both strands, and when they try to fix it, it doesn't work very well, and it usually makes it non-functional. The other possibility is that you could instead use the template and enzyme for a specific repair. So you could instead cut it and then insert what they want to use instead for the repair, put a reverse transcriptase in there and get it to copy in the target instead. So that is a possibility. It makes it a little bit more complicated. So it's really good at knocking things out. A little bit harder to kind of work around with the repair stuff. Again, you don't have to explain what it looks like in bacteria. You do need to understand that you would have to make a guide RNA with a complementary strand to your target gene. You would have to understand that the Cas9 enzyme is needed to make the cut, that it finds it with the guide, Cas9 makes the cut, and then as a result, it is going to then either correct or leave mutated depending on the purpose. All right, I think that's all things. Good luck.